Hi everyone, in this video we will discuss orally disintegrating tablet special dissolution challenges. Orally disintegrated tablet or ODTs are designed to break apart rapidly in mouth using minimal fluid. Because of this unique design, their in vitro dissolution behavior is very different from conventional immediate release tablets. For ODTs, dissolution testing must reflect two key aspects: very fast disintegration followed by complete drug release. So the goal is not just speed, it's to ensure rapid breakup in the oral cavity and reliable drug release under standard lab conditions. ODTs require a different testing mindset because patient experience starts in the mouth, not in the stomach. ODTs must respond instantly to moisture, yet remain stable during manufacturing and packaging. That balance is challenging. The key characteristic of ODTs: very fast disintegration, typically within 30 seconds. Minimal liquid in mouth. Saliva is only 2 to 5 ml. Often include taste masking coatings, contain high level of superdisintegrants, have fragile mechanical strength. Their porous microstructure allows rapid saliva penetration, but that same structure makes them sensitive to handling, humidity, and compression force. That's why mechanical fragility directly affects performance. Too hard, slow disintegration; too soft, breakage during packaging. Standard dissolution volumes like 900 ml do not reflect real saliva conditions. In the mouth, saliva volume is only 2 to 5 ml, so lab test can never fully mimic in vivo behavior. Key challenges with ODT dissolution: saliva condition cannot be fully replicated. Rapid disintegration disturbs normal hydrodynamics. Test masking coatings can delay drug release, making ODTs appear slower than regular IR tablets. Floating particles interfere with proper sampling. Fine particles may stick to vessel wall or filters. So, what we see in the lab is often a compromise between physiology and standardization. ODT dissolution is not about copying the mouth perfectly. It's about ensuring fast, complete, and reproducible drug release under controlled conditions. Many people confuse disintegration and dissolution, but they are not same. Disintegration means tablet breaks into small particles, and dissolution means drug actually dissolve in solution. For ODT, they may disintegrate very fast, but dissolve slowly because of taste masking coatings. These polymers are designed to slow drug release in the mouth, to hide bitterness and improve patient acceptance. However, in the lab, the same coating can delay dissolution, give a false impression of slow release unless tested with proper hydrodynamics. So remember, fast disintegration doesn't mean fast dissolution. Release is controlled by the functional or taste masking coating. ODT fragments often float on the surface because of trapped air or hydrophobic coatings. These light porous particles can move erratically in the vessel, get trapped in the stagnant zone, settle unevenly, and cause coning at the bottom. That's why apparatus selection and RPM are critical. The wrong setup can give misleading results. The right setup ensures realistic and reproducible dissolution data. So always observe particle movement, optimize RPM, choose the right apparatus. Good hydrodynamics means reliable ODT dissolution test. High porosity ODT fragments often float on the surface, which can distort dissolution results. In such cases, using a basket improves consistency by keeping all particles fully immersed. However, for very fragile ODTs, be careful. Sinkers can crush or compress the tablet. This may artificially change disintegration and release. Apparatus selection matters. Apparatus 2 most common choice. Apparatus 1 best for floating particles. Mini vessels used in early R&D for small doses. Sinkers only when large fragments float. There is a scientific trade-off in ODTs testing. Physiological relevance versus analytical detectability. In real life, saliva volume is very small, only 2 to 5 ml. This cannot be realistically replicated in the lab. That's why in R&D we often use simulated saliva, pH typically 6.2 to 7.4, but in QC we use standard buffers, larger volumes, 500 ml to 900 ml, to ensure accurate measurement and reproducibility. So, small volumes means more physiological, large volume better analytical reliability. For ODTs, with the test masking coatings, the media must be realistic enough to reflect oral conditions, but robust enough for routine testing. During method development, always screen multiple RPM. Regulators expect you to choose a lowest RPM that still gives reproducible results. Why? Too low RPM, floating particles, poor mixing, and high variability. Too high RPM, overly aggressive conditions, masks real formulation differences. Typical working range is 50 to 75 RPM. So your goal is stable hydrodynamics, minimal particle coning, clear discriminatory profile. RPM must selected scientifically, not by habit. Common test masking and release controlling polymers include UWT, HPMC, and ethyl cellulose. These polymers coating delay drug release, control batting and diffusion, directly affect the dissolution profile. Hydrophobic coatings slow wetting, delay drug release. Film defect or thin areas, steep early release, risk of test breakthrough. That's why discriminatory testing is critical. During method development, test undercoated batches and test overcoated batches. Confirm the method can detect real differences. Remember, coating thickness variation changes dissolution curve, directly impacts F2 similarity. A good method must separate good from bad coatings, not hide the differences. Sampling techniques is just as important as method itself. For ODTs, many fragments float on the surface. So your sampling cannula must avoid this layer. Sample from the mid-zone, not the top. Filtration is critical. Fine particles can clog filters. Choose carefully 0.45 micron for most methods, 0.22 micron when higher clarity is needed. Always perform filter winding studies. Confirm no analyte adsorption. Why this matters? Suspended particles, uneven distribution, and sampling bias. Bad sampling equal to bad data, even with perfect method. Always follow a structured approach in method development. First, observe behavior. Pass the tablet or fragment in real time. Then adjust step by step. Use basket or sinkers for floating fragments. Optimize RPM 50 to 75 RPM. Improve degassing to avoid bubble attachment. Select physiologically relevant but robust media. Validate filtration early. Never guess, always observe. Real-time visual monitoring is the fastest way to diagnose problems and design a reliable method. Observe, adjust, and validate. To conclude, ODTs are patient-friendly but analytically complex. They disintegrate very fast but dissolve variably. Key challenges include test masking coatings, floating fragments, irregular hydrodynamics. That's why operator selection matters. RPM must be optimized, media must reflect real behavior, filtration needs special attention. Robust ODT methods don't happen by chance. They come from careful observation, smart optimization, and validation. Good science equal to consistent performance plus regulatory confidence. Thank you for watching. In next video, we will discuss fixed dose combination, dissolution strategy, and multi-activities. Please subscribe us for getting update on upcoming industry application oriented videos and stay tuned because in this 62 video series we will cover dissolution method development, validation, troubleshooting and many real industry example. Please share your queries in comment section. See you in next video. Thanks.